So at this time, I'd like to introduce Dr. Elizabeth Bick, who is a Dutch-American microbiologist who has worked for 15 years at Stanford University and 2 years in industry. Since 2019, she has been a science integrity volunteer and occasional consultant who has scanned biomedical research and literature for images and other data of concern and has reported over 5,000 scientific papers. As you all know, research integrity problems has been a significant issue with COVID-19 and has created abundant misinformation with the public. So at this time, I'd like to turn the floor over to Dr. Elizabeth Bick. Dr. Bick.
Thank you so much, Kevin, for this introduction. So I'm both a microbiologist. I've actually overlapped, I think, one day with Dr. Maiwald here in the lab of David Relman at Stanford. And the work that I did there was on the microbiome of humans and marine mammals. So basically investigating which bacteria inhabit our houses, our bodies, our guts, our mouths, and other body parts. But I got interested while I was at Stanford to work on research integrity. And I later worked 2 years in industry. But since 2019, working on science integrity is my full-time job. So the work I'll be presenting today is mostly about that. I do run or have founded 2 blogs. So one is still from my microbiome days, Microbiome Digest, and that's a blog that is currently run by volunteers. And that's just the latest paper in the field of microbiome. And I now run Science Integrity Digest, in which I don't post very often because it's a lot of work to make a nice story. But you can take a look there and see the types of things that I have been working on. I'm also very active on Twitter, where you can find me on Microbiome Digest without the E, because Twitter only handles so many letters in their Twitter handles. So before I start my talk, I do want to make sure I have all my disclosures out in the open. So I make my money by consulting. I'm not employed currently by any university or by industry. I'm my own boss, and that's really great. I enjoy that very much. So the work that I do as a consultant is that I might be hired by universities or scientific publishers to work on cases of misconduct. So for example, if a university receives allegation of misconduct, then I might be an expert looking at, for example, images that look like they might have been duplicated or photos that might have been manipulated. And I might, for example, look at originals provided by the authors and see if those match the published papers. I also get speakers honoraria to give talks. Again, I'm not employed at a university, so this is one of the ways I make money. I did work 2 years at a company called Ubiome, where we sold kits to clients to have their microbiome determined, similar to a 23andMe kit, where you would order a kit, you would get a box at home, you could sample your poop, and then we would send it back to the lab, and then the company would sequence the genomes of these bacteria and tell you which bacteria you had in your poop sample. And part of the company also was selling these kits to insurance, medical insurance. And so unfortunately, the founders did make some bad decisions. And so they have now been charged with insurance fraud. I already had left at that time, but I did work at a company that is now involved in insurance fraud charges. But the FBI never talked to me, so the founders have been charged. None of the other employees have been listed in these charges. But I do have 4 patents from that time. I don't think they're worth anything, but it is something I will carry with me for the rest of my life on my resume. I also have a Patreon account where people can donate small amounts of money every month to support my work. And I have quite some supporters there. And that is also now generating a source of income for me. And it allows me to work on the cases that I want to work at. So I am a consultant, but I don't want to spend full-time consulting. I want to have most of my time free to work on any case I would like to work. I also would like to acknowledge many other people who work or who do similar work as I do. So I've listed a bunch of them here, people I've worked with. Most of these people are active on Twitter. And these people work on criticizing or at least finding errors in scientific papers and discussing those. And those errors could range from plagiarism to statistical errors to image duplication, which is the part that I'm working on. And some people also provide the platforms that we use to post these issues. And so I want to acknowledge all these people, and many of them work under a pseudonym because we do criticize other people's papers. So you can imagine that is a risky business. I work almost always under my full name, but some people choose anonymity, and for good reasons. So why do we care about scientific papers? Because you might think that a scientific paper, once it has been published, is set in stone, right? It has been peer reviewed, an editor has looked at it, it has been carefully edited by the authors, it should be close to perfect. But that is not always the case. Even after peer review, a science paper can still have errors or can even contain fraud, which came as a shock to me because I always have believed that science is about finding the truth. And we try to figure out how cells work or how the human body works or how chemistry works. Like science for me is about finding the truth. And when I first heard about scientific fraud, I was really shocked and I didn't believe that was happening because most scientists are honest, but science as a field is not immune to fraud. There is fraud in science. So when there is a publication, a paper that contains either an error or fraudulent data, I think that's worth discussing because I see these publications as the building blocks, the way that scientists communicate with each other. We build upon each other's work. We cite other people's papers. We hope our papers, once they're published, are being cited. And publications for me are like bricks in a wall, the big wall of science. We layer these layers of bricks on top of the older papers. We build our work on the work of others. So again, if one of these papers, one of these building blocks, one of these bricks contains an error or fraudulent data, we should be able to discuss that. We should be able to raise these issues and maybe correct the literature once we have found an error or even fraud. In the United States, science misconduct is defined as one of three things: plagiarism, falsification, or fabrication. And this is a definition by the Office of Research Integrity, which is the office that oversees NIH-funded research. So plagiarism, most people are familiar with. This is copying other people's words or maybe ideas without giving credit. And this could be like a verbatim text reuse, for example. Falsification is where people do an experiment, but they change some of the data. Maybe they leave out an outlier. Maybe they raise a value so that it crosses a threshold. So a negative, if you change a couple of numbers, maybe just becomes a positive. Well, fabrication is where a researcher completely makes up data. So not even does an experiment, but just types in some numbers on their computer and makes a beautiful graph and publishes that, but did never do any measurements. I also want to say here that I think that behind every misconduct case, there's a sad story. So it's very easy to point fingers when we see a fraudulent paper and we might make fun of an image and we might make fun of people. But I try not to do that because I want to be respectful for the authors on these papers. And we don't always know who was responsible for the misconduct. I always imagine the following scenario of a bully who is a professor, a research leader, maybe the star of the university, but maybe not a very pleasant person, maybe a person who's very demanding, who demands that his or her staff or graduate students produces positive results. Maybe you might even imagine that this junior person, this graduate student, is on a visa, and if they don't produce positive results, they might risk being fired. And that might mean that they have to move back to their home country. Maybe they even have a family within days often. These are the rules in the United States. And so if you imagine that and then picture that this junior person maybe does some photoshopping or changes some data in order to get a nice positive result and maybe a publication and that could help them further in their career and save their job. And that is understandable. I sort of feel almost sorry for these people. And who is responsible then in that case? Is it the person doing the photoshopping? Sure. Or is it the person who created this atmosphere in the lab that is an atmosphere of bullying? And so it's not always clear who does that. I want to be respectful, and I'll show you some examples of what I think might be misconduct, but I don't want to make it about the persons. And so if you think about things that can be wrong in scientific papers, there's just a whole list of things. And I've listed a couple of them here, but it's a sliding scale because some of these things are small errors. Some of these are maybe bad decisions, like bad research practices, and some of these might be completely misconduct. And there's probably no such thing as the perfect paper. Maybe there's no such thing as a completely fabricated paper. There's probably a lot of gray value, a lot of varieties in between. And so I do want to focus about images. That is sort of what I am most known for. But I did start my career working on plagiarism. So when I first heard about misconduct, what I did is use a sentence that I had written in a scientific review paper in 2009, and this was around 2014. I put that sentence in quotes in Google Scholars, scholar.google.com. You go there, you put this sentence between quotes or any other sentence. And normally you would expect, as a sentence is like 7 or 10 words or so, usually it becomes unique very quickly. But in this case, I happened to choose, just by luck, a particular sentence that not only gave me my own paper, but also 2 other papers showed up. And one of them had been published, and one of them was like some predatory publisher, not a real paper, I guess. But there were 2 hits. And if you look at this particular paper, that was a paper written by a completely different set of authors 3 years later after I had published my paper. And so they had stolen this paragraph that I've marked here in red. That was my paragraph. They had stolen this particular set of sentences, almost verbatim. And not only that, they had stolen a bunch of other sentences from other authors, other sources. So if you mark this whole paper from 2012, a lot of the text that is marked by me with colors came from different sources. It was this patchwork of different sources that they used to a Frankenstein monster, if you will, like to create this whole new paper. And so I found more and more of these papers. It became this hairball of sentences that were stolen by multiple papers. And in the end, I had after a year of work, and I did this as a hobby. I still worked full-time at Stanford, sort of in the weekends and at night. I worked on this project, and I found about 80 review and some research papers, reported these to the journals. Most of these had 50 to 90% plagiarized text, not just one sentence. I don't care about that, but complete paragraphs stolen by other sources. And about half of these got retracted and some were corrected. But the other half was not addressed, unfortunately, and is still out there. So by another stroke of luck, there was this PhD thesis that I looked in, and it had a plagiarized text. That's why I was working on it. But it also had some figures, and those had been published in the end, not just in the PhD thesis, but also as chapters in a journal. And so I found this particular blot. This is a Western blot. And if you don't know what a Western blot is, that's not really important, but you can see it has bands and it has this little dot here that stuck out to me. I remembered that. And I saw the same blot in another chapter in this thesis, and it had the dot here. And then I saw it a third time in the second chapter, but now it was flipped. So I flipped this image, mirrored it, but it was the same image. So there was this one particular photo, this one protein blot that had been used 3 times in different orientation and cut out differently, cropped differently, mirrored, etc., to represent 3 different experiments. These are 2 time series, and then this is a different concentration, doesn't really matter. But these were 3 different experiments, but it was the same photo. And so that's not good. That's an inappropriate duplication, as I called it. So these 3 PhD thesis chapters had been published as scientific papers, and I wrote to the editors-in-chief of the papers, of the journals, and they got fairly quickly retracted. And then I just thought, wait, this is weird. I guess I can remember this little dot. Maybe I have some talent for recognizing these blots. And immediately, maybe I'm too much of a scientist, but I thought, hey, how often does this happen? These papers have been published, they have been peer reviewed, but I guess nobody saw this or noticed that or paid attention to it. What would happen if I go online and I check more scientific papers for these types of problems? Would I find more of these duplications? And I did, and I also realized that photos in scientific papers are often unique. We can tell these photos apart. So if you look at these photos, these might be photos that you find in the scientific papers, you can tell that they're all different. And these photos are totally fine. There's no duplication here. But you can see this is a gel, like you can see all these bands are slightly different. These are bands in a protein blot, a Western blot. And so they all have different shapes and there's different backgrounds and stripes and scratches. There might be photos in research papers that look like nanostructures under a microscope or tissues or cells, and we can tell them all apart. And some people are better in that than others, but I guess I had some talent in seeing these duplications. And so as I was thinking about duplications, I realized there were different types of duplications. So I made these examples with photos of palm trees, which are not typically found in scientific papers, but this is just to illustrate this. And I found these photos on Unsplash, and I want to thank the people who make these photos available, because you can play with them and whatever you will, whatever you like, as long as you give credit. So thank you, people who post these photos on Unsplash. So on the left, I have what I call a simple duplication. So here are two sets of photos, and you can see that in, let's call this figure 1 and figure 2, we see the same photo of a sunset being reused. And if these photos are representing different experiments or maybe different tropical islands, that would be incorrect, because we can see and we can recognize that these are the exact same photos being used for two different purposes. So this is a simple duplication. Moving on to the middle part of the slide, we see a duplication with a repositioning. So we see two sunsets and we see that these photos overlap. So it's not just exactly the same photo, there's a shift. And maybe if you think about cells under a microscope, you can imagine that you move your sample under the microscope a little bit and you have a slightly different view, and that would be a different photo, but we can see that there's an overlap. So these are repositioned photos. You can also think about a photo that has been rotated or maybe flipped, mirrored, etc. That would also count as a duplication with repositioning. Looking at the right example, I have here photoshopped myself a set of two palm trees. And so you can see, you can recognize that part of this photo appears to be duplicated. So this would be a duplication with an alteration where part of the photo itself has been altered. And so you can also think about cells being photoshopped, maybe to make it look like there's more cells in a particular photo or fewer. And so this is a category 3, duplication with alteration. And if you also think about the types of duplication, you might maybe agree with me that the simple duplication is the most likely to have been caused by an honest error, maybe sloppy, but errors can happen. While the category 3 is very likely to have been done with the intention to mislead us, like these things don't happen by accident. Somebody actually photoshopped this photo. And so these types of photos can also happen in scientific papers. And so typically they're not photos of palm trees, they might look like photos like these. And so these are photos of cells in this particular example. And here's a simple duplication, and it might take a while for you to spot it, but these two panels here at the bottom left have been duplicated. They're exactly the same. And so if you look at the labels, they should show something different. Doesn't really matter what it's supposed to show, but in this case, these photos are different. All these photos look similar to each other, but these two are the same. And so I wrote to the journal and it got corrected in less than a year. It still took a long time, but I think this is a good outcome. I think this is just an error. Somebody just made hundreds of photos maybe and used in this particular case, the one that was labeled wrong or so. It's sloppy again, but it's not probably misconduct. This is just a sloppy error. This can just happen, and it got corrected, and I think that's a good outcome. Now here we're looking at a duplication with a repositioning, a type 2 duplication. Again, you see a bunch of panels here. These are cells under a microscope. Some have fewer cells or more cells. And you can see from the label that each of these are representing a different experiment. So all of these should look different and should be unique. But if you look a little bit more carefully, and maybe you might have spotted the duplication here, there are some panels that are overlapping with each other. For example, these two overlap, and I usually mark this with these colored boxes. So you can, for example, see an overlap here between these two panels, even though they represent different cell lines. And so this is a lot of overlap. If there's one overlap, I can imagine maybe this was an honest error. If there's this many overlaps, I would not trust anything else from this particular lab. That's just a lot of overlaps. That's just so sloppy that, you know, how can you trust anything else from this lab? And so this paper, I reported this in October 2015, and in March 2019, which is still a lot of time, but at least the paper got retracted. So retraction in scientific literature means it's watermarked as a retraction. It still should be available for you to read or to cite even, but it should be clearly marked as a retracted paper, basically saying you cannot trust these results. But it got cited by 27 people who didn't see this and might have based their research on this paper. That's not a good thing, but at least now the paper has been retracted. Here's another type 2 duplication with a repositioning, and this is a Western blot. So you see a bunch of panels here. And if you look carefully, you might see that these two panels seem to be showing the same sample, but in this case, it's flipped vertically. So these are like sort of mirror images of each other. They're also stretched slightly differently, but they're representing two different proteins. And here at the bottom, these two panels, these two GAP-DH panels, also appear to look identical. In this case, they're stretched vertically, slightly different. This paper has been cited by other papers 86 times. I reported this in October 2019, but unfortunately, it has not been addressed. And so this paper is still out there with its duplication. And yeah, I'm not sure, like, how can we tell the world that there's maybe some problem with this paper? So this is a special paper. It's worth mentioning because this is a paper from a lab by a pretty famous microbiologist in Marseille in France. And this microbiologist, this doctor, published a paper about hydroxychloroquine. And this paper came out in, I believe it was March 2020, so right when the COVID-19 pandemic started. And he claimed that hydroxychloroquine could cure all COVID-19 patients. But it was a very small study. It was only based on 20 people. And I had a lot of criticism about it. And I wrote a whole critical takedown of this paper. And I also thought maybe it would be worth checking some other works by this research group. Because, yeah, there seems to be a lot of problems in his papers. And he actually is a person who publishes a lot. He has published over his whole career 3,500 papers, which is just unbelievable. That's just so many. Like, it would be the equivalent of a movie director producing 3,500 movies. Like, you would not believe that a person can have that much work done in their life, because every paper is almost like a movie. It might take a year of your life to get it published. And so this particular image caught my eye. It's a Southern blot. It was cited by 116 papers. It's a microbiology paper. But if you look carefully at this image, there's parts of this photo that appear to look duplicated. And I've marked them here with colored boxes. For example, this smear here looks the same. And this part marked here in blue also looks the same. And some of the bands look remarkably similar. And the longer you look, the more of these repeats I saw. And so this famous microbiologist, yes, he has published a lot, but he seems to have cut some corners here in pursuing the truth. I don't think this is a very truthful image. It's very hard to imagine a technical error that could cause these duplications. And so I believe this image might have been photoshopped. And there has been many more papers from this particular research group that have now raised concerns. And so maybe we cannot trust all these 3,500 papers. Now here's another type 3 duplication. So these are duplications within the same photo, like the ones I showed you previously. And this is one of my favorites. This is a photo of nanoparticles. So this is like scanning electron microscopy photo. So basically looking at really, really tiny structures under a very magnifying microscope. And so this photo, the longer you look, the more repeats you might see. And here's a photo where I have marked all of them. So there's a lot of duplicated parts, and I haven't even marked all of them. I did wrote to the editor in September 2019. They did not want to retract, but I've been writing back and forth. And I think by now I have hopefully convinced the editors that this is a photoshopped image. This is not pursuing the truth. This is not how science should be done. And so hopefully now the paper will be retracted, because I would not trust anybody who thinks that this is a scientific image. So I did this type of scanning of scientific papers, again, first as a hobby, and now I do it full time. But back in the time that I published this paper, I still did it in my free time. And I worked together with Arturo Casadefal and Frederick Feng, also microbiologists like myself, but also editors-in-chief of journals. So Arturo Casadefal was the editor of MBio, the journal in which we published in the end this paper. So it's a little bit maybe not completely properly done when you publish in your own paper, but we made sure it was peer-reviewed independently, and it was also later cited a lot. And I feel that nobody else wanted to publish this paper because it was so controversial at the time. And then Frederick Feng was at the time the editor-in-chief of Infection and Immunity. And so what I did is I scanned 20,000 papers from the biomedical literature by eye. And when I found these duplications, there had to be duplications within the paper, again, in photographic images. And I made sure to include papers from different journals, different publishers, different time periods, and a period of 20 years. And in that set of 20,000, I found 800 papers to contain duplicated images. So that's 4% of these papers. And both of my co-authors had to agree with my findings. So if I found a duplication, both of them had to agree. If one of them did not agree or thought it was maybe not duplicated, or maybe there was a perfectly good reason for it to be duplicated, then it was not included in that set of 800. And then we sort of estimated that about half of these papers, these duplications, had been done intentionally. So of course, that is hard sometimes to know for certain for a particular paper. But if you have a set of 800, you can make sort of educated guess. So we estimated about half of these papers, so 2% of all papers that I scanned, to have intentionally duplicated images. So that would be misconduct. And the other half might have been honest errors. Now, does that mean that all, if you think about all scientific papers, is 2%? Can you extrapolate that to all scientific papers? What is the real percentage of misconduct? And I think it's actually higher than 2%. So I only looked at photos, and I can recognize duplications, but I know I probably missed a lot, because how many images can I recognize? I only look for images that might have been duplicated within that paper, but you can think about images that were duplicated across papers, or images that have been really well photoshopped and I might not have recognized it. I only can find the tip of the iceberg, the people who really make the dumb mistakes, who leave traces for me to find. And so if you also think about other types of images like line graphs or tables, research misconduct in those types of images would be even easier to do and harder to catch. So I think the real percentage of misconduct might be much higher than 2% and is actually in the 5 to 10% range. And that is a scary number. I also had some data now. I had this data set, and I could try to make a correlation between the impact factor of a journal and the percentage of problematic images. And you can sort of see this inverse relationship where the journals with a high impact factor, so like Nature and Science are here on the right, have a lower percentage of these paper problems than some low impact journals that can go up to 12% of their papers with images to have these problems. But it's not completely true. I mean, there's some journals that do really well, even though they have low impact factor. So it might have been more correlated to the editor or the audience or the type of papers that these journals publish than the actual impact factor. And it also gave me a data set to look at what happens once you report these papers. So all of these roughly 800 papers I reported to the editors in around 2014 and 15. And waiting 5 years, so what happened 5 years later, so this was last year or so, 65% of these papers had not been corrected or retracted 5 years after I've reported them. And I sent some reminders as well. So that means that 2/3 of these papers are still out there. And the percentage is still getting a little bit better, but yeah, about 60%, I think now we're getting close to 7 years. 60% of these papers are still not corrected or retracted. They still have their duplications. And yeah, that's frustrating, right? Because I try to alert the editors and they're doing nothing. They seem to be looking the other way. And I started this project sort of very naive, maybe, that I would change the world and have all these papers corrected, but that didn't seem to happen. By now, I've actually found, so this was already 2 months ago, close to 6,000 papers now that I have found, and about half of them are reported to the journals. And because I just find more joy in finding these papers than reporting it, but it should be reported. I should tell this to the world once I found a problem like that. So officially, when you find a problem like in a paper, any problem, image or whatever other problem, you should contact the editor-in-chief of the journal and maybe even the research integrity officer of the university where the research has been done. And as we have seen more often than not, these things are not being addressed. Investigation might follow, but in 2/3 of the cases, these papers are not acted upon. So I'm not doing it the official professional way anymore. I'm starting to post these things online. So I'm using a website called pubpeer.com, and I post all of the problems I'm finding on that website. So if you go to Pubpeer, you can post some concerns about a particular paper. Each paper gets a new page, and you can post all the concerns, and you can join a discussion, and you can post anonymously or under your full name. And I usually post under my full name. You can also install a plugin that will work with your browser or even with your reference manager. So if you do a literature search, so I've shown you an example here. I use PubMed to do a literature search on something. And then if a paper has a Pubpeer entry, it will look like this. It will get a green banner and I can click on it. That will take me to the website and I can see what people had to say about this paper. And I feel by posting these papers on Pubpeer, I can actually tell the world that there might be a problem with it. That's the best I can do. And I feel this is sort of now my new mission. Just flag as many as I can and share that with others on Pubpeer. So I've told about a bunch of problems that I have shown in scientific images, but there can be many other types of problems. And this is just a slide or a collage of some of the more funny ones. And I didn't work on all of these myself, but there are also examples of people whose name have been put on a paper that they weren't an author of, but their names are used by others, maybe to lend credibility to the paper. And they had no idea, and it wasn't really a good paper. But what do you do if your name is put on something and you don't want to be responsible for it? There are people who have promised each other to peer review each other's papers. And there's even complete fake peer reviews where people say, oh, here are 2 email addresses of 2 people who can peer review my paper. But these are Gmail addresses set up by the author, him or herself, basically allowing the author to peer review their own paper. And of course, it's really good. And it gets, a thumbs up. And so the people are very creative to get their papers published. And if you enjoy that, I would encourage you to read Retraction Watch, which is a blog that talks about retractions and the reasons behind them. And sometimes they're funny and sometimes they're just sad. And my personal favorite is one particular guy, I won't mention his name, but he's publishing and he claims to work at the California South University, which is a university that doesn't exist. He claims it's in Irvine in California, and there is a university in Irvine, but it's not the California South University. It's a completely made up university, and he has made an elaborate website, but it doesn't exist at all. And he publishes under this name or under this affiliation, and I'm not quite sure why, but it makes you laugh. Like, people go extreme ways to get their papers published. And then there's also complete organized crime, fake paper factories that make fake science, that publish fake science, and those are called paper mills. And so scientific paper mills are the organized crime, the organized science misconduct in the scientific publishing world. And these organizations, they have different business models, but basically they want to make money, and authors need to have papers published. And so this is a sort of a creative way of making money. So either they work with papers that have been accepted already, and then they sell an additional author. So basically they say, oh, we'll squeeze another name in and you have to pay for that, even if you didn't write the paper. Or they sell completely fake papers written by ghostwriters with fabricated data. Here are two papers from a journal, and the paper on the left was written by a particular group of authors. The paper on the right was written by a completely different group of authors, no overlap, but there were some images in it that were exactly the same. These two images, you'll have to believe me, but these two images are actually showing the same specimen. It's just upside down. And there were some other images that all these papers had in common. And in the end, I found 125 papers that all had shared photos, but they were supposedly about completely different topics. And so here's another set of papers, two papers that look sort of similar, but they all had the exact same background in these blots. And in the end, we found 600 or 700 papers that all had very similar plots. And we believe these are artificially generated, completely fake data based on all kinds of errors that they make. And they are getting retracted now. The authors don't even fight for their papers. They'll just say, oh, sure, retracted, because they have their paper published, I guess, and that's what they needed for their career. So there's certain countries that have very strict rules about authorships, and they have very strong incentives, and they almost force people to make fake papers. And yeah, that's very sad. And so it's driven by incentives and incentive structures in particular countries, how to publish. And so this is, I think, one of my final slides. This is a paper where you can see it's fake data because they made a big error. So this is a paper claiming to have worked on prostate cancer. But if you look at the gender distribution of these patients, it's very unusual. You would not expect half of your prostate cancer patients to be female. And so this is where you can see it's a template that they use over and over again. They just changed the disease and the molecules a little bit, but in this case, they forgot to take out the females. And so finally, I have been threatened twice for the work that I do by some legal threat. So one was our French friend, the doctor who claimed that hydroxychloroquine worked. He filed a complaint about me for harassment and blackmailing and all kinds of nasty things. He claimed I was doing that in Marseille, but I have not seen any real official legal action there, but it was scary. And luckily, the scientific community helped me by setting up a petition and showing their support. Because basically what I'm doing is, yes, I'm criticizing other people's work, but I try to do it scientifically, like I'm raising scientific concerns. And I don't think that these scientific concerns or discussions should be held in the courtroom, but people want to maybe silence what I'm doing. And these legal threats could have a chilling effect on scholarly criticism. So it was even written about in Science and Nature. And so this is my final slide, and I hope to leave you with some good thoughts and maybe some pessimistic thoughts as well. So I have talked a little bit about the percentage of misconduct. So it is, to the best of my knowledge, my guess would be it's about 5 to 10% of scientific papers contains data that either has never been measured or that has been changed and made to look better. And why do people do that? I've talked a little bit about maybe the pressure to publish or the bullying professor. There's all kinds of reasons, but basically scientific publishing focused too much on publications as a measure for productivity. And I know that in the Netherlands, where I'm from and actually presenting from today, there are new thoughts about maybe measuring the productivity of a researcher in other ways than publications in high impact journals. Maybe the work on, for example, peer reviewing other people's papers should be also counting towards their career achievements. And so there should be new ways we think about how to measure that. And also we should allow people to also publish negative results. If you've done 5 years of research and you've proven that your drug does not work on a particular disease, maybe that's worth publishing as well, because that would help other people not to reinvent the wheel. And we should be able to publish that. But unfortunately, most journals don't want to accept that. They only look for novelty. So there are all kinds of conflicts of interest. So scientific publishers seem to care more about making money than correcting the scientific literature or doing quality control up front. And institutions similarly, they care about their star researchers a lot because they bring in money, grant money, and they might not want to really investigate allegations of misconduct. They might want to look the other way, and there's too many things that are being swept under the rug, and we should change that. But whose role is it to detect science misconduct? So I'm a patron-supported volunteer doing this work, but it's not really a paid job. It's not a professional job. And it's also a dangerous job because it sets me up for legal issues, shenanigans, and it could financially ruin me. The way that things work in the US, you can be completely right and you can still be sued. And that will cost you money to defend yourself, even if you're innocent. And that is bad. And that is, a situation I'm well aware of. And it's at some point could, tear me apart financially. And I'm still willing to take that risk, but it is a scary situation. And then finally, there's a tremendous cost of science misconduct. All these scientists might replicate or try to replicate these fraudulent data, but on an even bigger scale, science misconduct gives science a bad name. And we already have seen in the past 2 years, all the misinformation and the loss of trust in scientists. And you could walk away from my talk saying to anybody else that all science is flawed and we cannot believe in science anymore. And I don't want that to be the message that you take home today. I do want the message to be that we should trust in science, that we need science to solve our big problems that we have, and that science, yes, science has flaws, but we can make it better. And there's people, not just me, but there's many other people who care about the quality of science and we want to make it better. And that should be the real message, a message of hope. So I do hope I have made you a little bit more cautious in interpreting science, but also have given you some hope that there are people caring about these issues. I do play a game on Twitter, so you can follow me at Microbiome Digest. And people love it when I play this game called Image Forensics. So I just make it about finding the duplication. So you might look at this image and you might spot a duplication here. And the first person to have spotted the duplication will get an emoji award. And there it's just a fun game that I have. So with that, I will stop sharing and I see there's some questions. So I'll be happy to take those. Thank you so much.